Daniel has been with DREAM from the time he was a student, and some of his work was submitted as one of the first, one of the algorithms that won in the first DREAM challenges. And since then, Daniel has been very active in the DREAM challenges. So I was very pleased when he suggested this challenge that he organized pretty much on his own with his team at the University of Lausanne. University of Lausanne, right? Yes. So I leave you with Daniel and the disease module identification DREAM challenge.
Hi, everybody. Thanks for coming. So it's a pleasure to present this challenge to you today. I should say this is really work together done with Sarvena Shuptar. She co-led the challenge with me and did actually the bulk of the work. Unfortunately, she could not be here for the conference. So I will just give a very brief review of our previous work that will just be 3 minutes as a motivation of the challenge, give an introduction to pathway network analysis, and then present the challenge setup and some of the results. And afterwards, we'll hear from 2 of the best performers. So we now know many genetic variants that are associated with complex disorders, but progress to develop personalized or targeted treatments has still been slow. One of the reasons is that we still don't understand well the mechanisms in between here. Of course, genetic variants are sensed and propagated by molecular networks. The picture is even more complicated if you think that actually these networks are actually rewired differently in each tissue. And so in this previous work that I did, we constructed a collection of close to 400 cell type and tissue specific regulatory circuits. So that's the largest collection of such fine-grained regulatory networks to date, connecting transcription factors, regulatory element, and target gene isoforms. So I don't have time to present this method, but this is published. And I just wanted to highlight one result, which was kind of the motivation for this challenge. So one question we asked was whether disease-associated genes, so genes that are perturbed by genetic variants that are associated to the diseases from GWAS data, if they are clustered in these networks. So do they interact more than expected? And so I developed a pipeline where you can kind of measure this over the whole network to have this kind of an enrichment score for the network. And so we did this for all these 400 networks. And if you see, for example, for schizophrenia, you indeed see that we have a significant enrichment for clustering of these disease genes. And especially interesting is that it's for brain tissues. And what was really surprising that even more fine-grained, you get the strongest signal in this case for structures of the basal ganglia, which modulate cognitive and emotional behavior and are actually the primary target of antipsychotic drugs. So this suggests that really the GWAS data can pinpoint regulatory modules in disease-relevant tissues. But we didn't yet identify the modules here. So that's why the next step, that is just a global enrichment score here. So the next step was really to identify the pathways, the modules. Schizophrenia was just an example. We had such results for many different traits and diseases. So as I mentioned, this is published. So what is a module or a pathway? So we use these 2 words interchangeably here. It's a gene set, module, pathway. It's a group of functionally related genes or proteins. So some traits are just defined by a single gene, but most common diseases and biological functions of interest, of course, involve multiple genes. And we've seen yesterday and through this conference many talks presenting pathway analysis in one form or another, because it's a very common problem in molecular biology and biomedical research. We have some omics data. We want to know what are the pathways really underlying our disease or our biological process of interest. And so one approach is pathway analysis, where you start from pathways from a database, such as gene ontology, KEGG, and so on. You test these pathways for enrichment in your data. Well-known example is gene set enrichment analysis, which was presented by Jill yesterday. Limitations are, of course, that this is limited to known pathways, and our knowledge, of course, is still largely incomplete, and also these databases largely lack tissue-specific information. So an alternative approach is network analysis, where you build a network from your data, and then you identify modules in these networks. So that's module identification. And then you can analyze the modules and maybe bring in pathways at this point to annotate your modules. An example is weighted gene co-expression network analysis, not my favorite method, but it has a very nice R package. So it's a good place to get started. So the advantages of this approach is that we do not rely on known pathways, so we can discover novel pathways, expand known pathways, and also network data is now becoming increasingly available, so this becomes more and more relevant. So we had previous challenges focused on network inference, right? But here now we were really interested in module identification, and this is really a classic problem in network science. There's hundreds of methods, so really ideal for a dream challenge because there's a large interested community. But the problem, of course, is it's really tricky to know how to assess, how can we assess methods, which are the methods that are best to identify biologically relevant modules. And because in the literature, the predominant approach is to use artificial benchmark networks using stochastic block models, for example, where you have an edge probability matrix where you can really generate artificial networks with a predefined modular structure and then see if the methods recover that. So there's different benchmarks that are widely used. You can try to make them more realistic by having different module sizes and so on, but of course still this doesn't look like true biological networks. So you can use real networks, and for some small networks, sometimes there's some benchmark networks where community structure is known. That's of course not the case for biological networks. So we can use metadata that correlates with community structure, like geo annotations or pathway databases. And you see this pathway is enriched for a geo term maybe or pathway, so we can say it's supported. The problem again is limited to known pathways, and also often these pathway databases are not truly independent from the data that was used to construct the network, because maybe the network uses protein interaction or gene expression data, which is also used to annotate genes. So it's not truly independent. So we had this idea to use GWAS data, which is really a novel approach for module assessment that was not really previously used. And GWAS has the nice property that it provides kind of a genome-wide unbiased functional annotation. So the GWAS is an experiment where you get at every genomic location, you get the association of that region of these variants to the given trait or disease. So it's not biased towards well-studied genes or proteins, right? It's genome-wide. Also, it's not biased towards some cell types or tissues. It's independent. So, and this is completely orthogonal to the network data. So it's a good way to validate. So what we do, we collected actually the largest collection of public GWAS data sets to date, over 200 traits and diseases, and so these numbers are not up to date here actually, but we have many different types of traits and diseases, and now we can test modules for enrichment in GWAS data. So here you see this module is enriched. It contains more disease-associated genes than expected. So the GWAS, which is independent data, really supports this module. It supports that these genes are functionally related because they're all associated to the same disease or trait. Now, how to do that is actually very tricky. To do this GWAS pathway analysis. So we developed a tool called PASCAL, so that's published, was developed by David Lamparter and myself, and this tool allows you to very efficiently summarize GWAS data at the level of genes. So you have the GWAS summary statistics, which are p-values for every SNP, giving the association to the disease, and you can summarize that at the level of a gene. So I don't have time to explain that, but it's a difficult problem because the SNPs are correlated among each other. There's the linkage disequilibrium, so you have a correlation structure that needs to be taken into account. Now, at the level of modules, we can also use this PASCAL tool to, instead of single genes, compute the p-value at the level of modules. Here you have 3 genes, for example, and now something that I would like to stress, because it's often neglected in pathway analysis, is that genes that are nearby on the genome, they're not independent, right? They have correlated signal due to linkage disequilibrium here. But even if you use gene expression data, neighboring genes often have correlated expression. So neighboring genes on the genome cannot be treated independently when you do pathway analysis. Otherwise, your analysis, you will get inflation of p-values, you will get false positives. And often in the literature, you see that this is ignored and the p-values won't be correct. So I don't have time to show this, but you can look it up in our paper. the effect of it's really crucial to deal with these proximal genes. Okay, so one of the first analysis that Sarvenas did was she tested pathways for enrichment in these GWAS data sets from known pathways and then modules extracted from networks. And as you see, for some traits, shown in blue here are the known pathways. For some traits, we actually get a good many hits on known pathways, and this is too small to read, but these are immune-related traits. So it seems for immune-related traits, we have good coverage in the existing pathway database. But then for other traits, and that's especially, for example, psychiatric disorders, we get no hits in the known pathways. But we do get hits in the network modules. So that suggests that there's a big potential to discover novel disease pathways using the network-based approach, and was kind of the point where we decided we should really launch this challenge to crowdsource this and try to discover novel disease pathways in this way and to evaluate the best module identification methods. So the challenge has 6 networks that were contributed by different groups, and you see we have 2 protein interaction networks, a signaling network, a co-expression network, a cancer-related network, and a homology-based network. So they're diverse, different size also, different number of edges, and different structural properties like degree distributions. So it's a diverse benchmark collection, and they're all weighted, undirected, except for the signaling network. And they're all unpublished or custom versions of published networks. So this allowed us to anonymize the networks by removing the gene labels. So we removed the genes names and replace them just by numbers. So the participants only see this, so they cannot use any additional information. This was purely using network topology, go from topology to function. And yeah, I wanted to thank Casper because he put me in touch with several of these groups who provided the networks. So we had 2 sub-challenges. Sub-challenge 1 was classic module identification. You just run module identification on each of the 6 networks. Then sub-challenge 2 was multi-network module identification. So you have several, you could use all 6 networks which were aligned, or you can also think of it as a layered networks, and identify integrated modules across networks which potentially could be more robust or more refined in some way. So the submissions had to be a set of modules that was non-overlapping, so we do non-overlapping modules. The number of modules was not fixed, obviously. The size was constrained to at most 100 genes, and that's an important point that's a bit different from standard module identification that's often used also in biomedical research, is that these module identification methods often give you very large modules with like 1,000 genes, which is really not useful. So you often have like reasonably sized modules and then some very large modules. And that's because they cannot really have they don't really have an adaptive resolution, these methods. And here we said like, well, 1,000 genes, that's really not useful to gain biological insights or to guide follow-up experiments. So we really wanted that large modules are further broken down by kind of changing the resolution. So that was a bit of a unique aspect of this challenge. The score that was used to rank teams was simply the number of modules showing enrichment in at least 1 GWAS, of course corrected for multiple testing based on the number of modules. And also each module was only counted once, even if it showed enrichment in several GWAS. So we had a leaderboard phase where teams could make up to 20 submissions, a final phase with 1 submission per team. Of course, different GWAS datasets were used in the 2 phases, so an independent holdout set was used for the final scoring. So here you see the scores. You see that 2 teams achieved a score of 60. We also did a bootstrap analysis to kind of see define ties between teams, and we subsampled the GWAS datasets 1,000 times and computed the base factor. But basically, if a team is better or equal than the best team more than 1 out of 4 times in the subsampling, then it would be considered a tie. So on this plot, the difference look not very big among the top teams, but actually we looked also at different FDR cutoffs. So this is at 5% FDR. And we found that this one team, Team Tusk, will present after me, actually ranked first at every cutoff that we looked at, and also ranked first on the leaderboard GWAS that they said, and they also did well in sub-challenge 2. So they had a much more robust performance than some of the other teams. Well, some other teams also performed competitively. The differences are really not that big. So here you have this team Aleph who tied, and at other FDR cutoffs we have 6 other teams who also tied for second place. If you look at the different networks, it doesn't look that pretty anymore. You see a lot of variability, so not the same method performs best on each network. So we have to analyze this further to better understand the advantages and limitations of different methods, and this also suggests that maybe community predictions integrating these methods could maybe improve robustness. So that's things we are looking at now. In sub-challenge 2, so that was the multi-network predictions, you see that we have a tie of several teams. But here we also compared, as a baseline, we compared the multi-network predictions with single network predictions, because of course if you just take a single network like the co-expression network, which is the one that scored well here, so that's just from one team of sub-challenge 1, actually the one that performed best on the leaderboard. You see that they got the multi-network predictions didn't really improve the score significantly by adding these additional data networks, which was a kind of a surprising and interesting negative result. We are maybe these modules have other properties that could be interesting. Maybe they're different in some way or better in some way that's not apparent here in this score. So we're looking at that now, but just based on this score, we could not really say that they significantly improved over the single network, so we declared there was no winner, no official best performer in sub-challenge 2. So I would like to congratulate all best performers and also thank all participants. We'll have 2 talks that now, right now, of the best performer of sub-challenge 1 and also the team that achieved the highest score on sub-challenge 2. So I'm looking forward that they shared their insights on this challenge. And we also have several posters of participants, including 2 runner-ups that you can look at after the talks. So I would like to conclude with a final thought. So, how was the time? This analogy was actually from a TED talk that I saw that talked about where we often have these simplified representations that are used a lot, and then kind of we have these in our mind when we see a problem. And an example is here, you know, how does a plane fly? You all know this profile of the wing that generates the lift, but then that's not the complete story, because how could planes fly upside down, right, if that's the explanation? So I often use these drawings of modules and so on, and that's how we think of modules. But similarly, maybe we shouldn't just, we should maybe think out of the box and look at different types of models as well. The next talk, I think, will mention these dense bipartite modules that they also looked at. So basically, you could have different structures in these networks that could be relevant for disease, not necessarily just the densest clusters. And actually, Aaron did some analysis on this also and saw that just the modules with the highest modularity were not necessarily those that turned up significant. So just optimizing for this density of the interactions in the modules is not necessarily the only or the best strategy. So I would like to thank, first of all, Sarvenas, who really did co-led this challenge with me. Of course, the network contributors and participants, David, I mentioned, and of course the DREAM team and the SAGE team, who are really supportive, and also Vital IT computing cluster, because the scoring was very challenging, was very computationally intensive. So for the leaderboard phase alone, we launched over 400,000 jobs on the computing cluster. So that was a challenge, but so we're thankful for their support. So with that, I conclude. Thanks.
So we have time for 1 or 2 questions. So, you know, I have a question that you alluded to very much at the end of your presentation, that in a way, what is a module, right? I mean, there is, in the biology, there is nowhere where a module is, right? So it's kind of a figment of our imagination to some extent. So do you think that we can, going forward, define a module a little bit, following the same lines of Zach Cohen's presentation, define a module using your scoring technique?
No, I mean, I wouldn't use that to define what a module is. I mean, I think these modules or gene sets is really about, is it useful for your analysis, right? And if we want to identify genes that are that underlie, I mean, this scoring is really focused on the GWAS analysis, and I think that should be kept in mind. Maybe for another problem, another goal, maybe the best scoring method here would not necessarily be the best approach. So here we really saw which methods give the modules that give the best signal for this GWAS analysis. So I think that should be kept in mind, and I wouldn't use it now to define a module based on just this scoring metric. And we also, I haven't shown this yet, but we obviously want to look at other evaluation metrics to see how the methods do if you look at other approaches like modularity and structural properties and so on.
So to follow up on that point, and in agreement with Gustavo's question, you say that if the module doesn't have its intrinsic definition and is primarily for use in analysis, which I agree with, then in this case, the analysis being find a module with enriched for PPI interactions, and then ask if it's enriched for GWAS. Right. But then if I could say that I'll actually look at the GWAS while I look at the PPI, and have an objective function that enriches for both in some balanced way. And then I've redefined the module, and I'm not sure if people will, so if I didn't do it, I did it in a two-step way, then you will say, that, you will report p-values of GWAS enrichment on a module that was separately obtained and state it as a claim. Whereas if you combine those two types of enrichment into one objective function and find a module that enriches for both, it's no longer a claim because you searched for it. So this aspect of making a claim, rejecting a null hypothesis, by a second enrichment test is then lost. And in order to do that claim, you have to have more investment in the definition of the module. What is the claim if the module itself is not defined and is only useful for downstream enrichment tests, right? So it becomes important to have faith in the notion of the module as being intrinsically valuable, and then you can claim that it's also enriched in my GWASs. You understand?
So I think for a very interesting discussion, which we definitely should continue after the talks, because there's a lot to be said. But yeah, I think definitely. So there's a whole different class of methods that take an integrative approach that use both the network structure and the GWAS data at the same time. So that's another approach that is definitely also valid and could potentially find modules where we don't find modules here. But the advantage here is, well, one of the goals was really to evaluate module identification, the classic type of module identification methods. And the problem is when you add the GWAS data, we wouldn't have a way to independently score them. So for the challenge, it wouldn't work because we would have to find another way. So that was important here. We separated this, mainly to have an independent orthogonal way to score the modules.
Yeah, I appreciate the way you framed it. I couldn't
Yeah.
buy into it. I just wanted to follow up on Gustavo.
Do you want me to ask? Sure. Very quick. Okay, I'll be very quick. So all these GWAS analyses have these tissue relevance. So because every disease has a ramification in a particular tissue type. So I wonder if your design has some tissue specificity to it, or is tissue agnostic?
Yeah, no, so obviously, as you can see from my previous work where I did all these tissue-specific networks, I think this is really crucial to have tissue-specific networks in the end. So here the networks are not tissue-specific. They're kind of global protein interaction networks like string and so on that kind of aggregate all the information you get from different domains. But the goal is that these methods could be applied when you have your own data set and now you construct your network from your gene expression data, for instance, that's now tissue specific. You could then apply these same methods to get a tissue specific module. So it was not done in the challenge, but you could do it then with these methods on your own data. Yeah.
Thank you.
Thanks.
Thank you, Daniel.